Hello students, my name is Sonali. Welcome to our channel Success EduTech. Thank you for your kind support and response. It matters to us. Please continue to like, share, subscribe to our channel. Today we will see part 2 of the plant-water relation. Kindly watch my previous videos. Link is given in the description and press the bell icon. Now we will start our today's session. First we will see about water available to roots for absorption. Plant absorb water from rhizosphere. Rhizome means micro and spheres means environment. So the micro environment surrounding the roots. Now this rhizosphere is very important for regulating the soil organic matter and the decomposition and nutrient cycling, and it is important for the growth of the microorganisms also. Water present in the soil occurs as gravitational water, hygroscopic water, combined water and capillary water. Now, what is meant by gravitational water? I will explain from the diagram. It is a free water moving through the soil by force of gravity. It is largely found in the macrosphere of the soil and very little or no water available for the plant absorption. Means this gravitational water is passing from the soil macrosphere of soil particle and it will drains right through in the soil to form a water table. Okay, now the hygroscopic water means the water adheres to soil particles due to the force of attraction. So this water is also not available for the plant absorption. Now this hygroscopic water absorbed from the atmosphere and held very tightly by soil particle. So it is not available for the plant absorption. Now the combined water is nothing but the soil minerals present in the soil, that is silica, aluminium, and when it will combine with the water molecule, it forms the hydrated oxide like SiO2, then Al2O3. So this water is also not available for the plant for absorption. Now the capillary water is only available for the plant absorption. We know the meaning of capillary is the action of plant to take up the water into the upward direction. Okay, and that is done by the xylem tissue. So this capillary water we will discuss from the diagram that this water held in the microspores of the soil particle. See, these water molecules pass in the upward direction, and this water that remains in the soil after gravitational water is drained out, and the movement of water within the spaces of porous soil particles is known as capillary water. Now we will discuss about absorption of water by roots from the soil consists of the following three physical processes, that is imbibition, diffusion and the osmosis. We will discuss about imbibition. Imbibition is defined as it is a swelling up of hydrophilic colloids due to adsorption of water. Now hydrophilic means the water loving substances and the colloids means the substances, one substance will disperse into the another substance, for example milk or any other substance, butter. Okay, now we will see from the example. As suppose this is the resins and soak into the water. So after 1 hour, imbibition process will form. That is, resin will absorb the water and it will turgid and swell. Means this is the imbibition in resins. The meaning of imbibition is nothing but the absorption. And the first is the adsorption of water. Adsorption is the surface phenomenon. So the water will on the surface of the resin. So it is adsorption, and when it will enter inside the resin, then it is absorption. So here the imbibition process is complete. Now it has two parts. that is substance that absorb water or liquid is called imbibant, means absorbent. The substance that absorb water means here our substance is the resin, and the water or any liquid that gets imbibed is called as imbibate, means absorbate. Right. Now we will see the next process, that is diffusion. Diffusion means the dispersion. Now this diffusion is defined as the movement of the ions, atoms and the molecules of a substance from the region of their higher concentration to the region of their lower concentration. See from the example, here the solute molecule and in the disperse in the solvent molecule, that is here it is a high solute concentration, and when it will diffuse, when it will disperse evenly in the solvent molecule, diffusion process will complete while equilibrium. Now we will see, and this process is occurs due to the movement of their particles due to the kinetic energy of the molecules. So diffusion continues till an equilibrium is reached. Now diffusion is result in the diffusion pressure, that is known which is directly proportional to the number of diffusing particles, means the quantity of diffusing particles. So the diffusion pressure is related to the particles which will diffuse. Diffusion pressure is of pure solvent is always more than the diffusion pressure of the solvent in the solution. So the difference in the diffusion pressure of the pure solvent and the solvent in the solution is called diffusion pressure deficit or suction pressure. Now the term was coined by B.S. Mayer in 1938. Nowadays term water potential is used for the diffusion pressure deficit, concentration to the region of their lower concentration. Now diffusion is a significant in plants in absorption of water, minerals and conduction of water against the gravity, exchange of gases and transport and distribution of the food. Now next we will see about osmosis. Okay, now what is meant by the osmosis? It is a special type of diffusion of solvent through a semi-permeable membrane, and it has three types of solution: hypotonic solution, hypertonic solution and isotonic solution. I will discuss it from the diagram. Now see, osmosis has a three solution, that is first is, suppose we will see hypotonic condition of the solution. Now this solution has hypotonic solution has a solution outside the cell. This is our cell and the solution outside the cell is a hypotonic, means if it has a lower concentration of the solute relative to the cytosol. So the solute proportion is less in this side and solvent proportion is more. So the water or any liquid present here, means the solvent will enter into the cell and cell will absorb to that liquid and it will turgid and swell. So this condition is known as hypotonic condition, in which weak solution and the strong solvent is present. Now second one is the hypertonic condition. Here the water or the solvent is more in concentration inside the cell, and means it is have a higher solute concentration than that of the cell. Here the higher solute concentration is present than that of the cell. So cell will release the water into the outside of the cell and the cell will shrink. Cell will flaccid. So this condition is known as hypertonic condition, where strong solution or a weak solvent is present. Now last one is the high isotonic. This solution where there is neither gain or nor loss of the water. Means as we discuss in the hypotonic, there is a gain of water inside the cell. In hypertonic, there is a loss of water outside the cell, from the inside the cell. So in isotonic condition, there is nothing gain or loss of the solvent into this condition. So neither the cell will shrink or nor it will be turgid. Now we will see about the types of osmosis. So osmosis is of two types, exosmosis and endoosmosis. So here first we will discuss about the osmosis. See here the concentrated sugar solution means in the solvent sugar like solute like sugar is mixed, and here is the dilute sugar solution means the solvent like water proportion is more. So obviously the water from here will enter into this side where the solvent proportion is less. So through the partially permeable or semi-permeable membrane, the molecule will cross this membrane and enter into this side. This process is known as osmosis, and water molecule passes through this, not a sugar molecule will pass the water molecule. Now see the types of osmosis, that is exosmosis. It is the migration of solvent from the cell outside, and endosmosis, it is the migration of solvent into the cell. It causes turgidity of the cell, and exosmosis will cause flaccidity of the cell, means shrinkage of the cell. See, this is the first diagram where the resin will swollen, resin will turgid after absorption of the water. So water will enter into the cell. So this is the endoosmosis. Now this B diagram will here the solute like salt is present in the solvent. So it is the solution that it is the salt solution, and the water from the grapes will coming out into this solution. So the grapes will shrinkage, the grapes will be flaccid, and this is the exosmosis process. Now we will discuss about the osmotic pressure. Now the pressure exerted due to the osmosis is osmotic pressure, which is required in opposite direction. So as to means osmotic pressure is a pressure of the solution which is required in opposite direction, so as to stop the entry of solvent molecules into the cell. Means if we will discuss about, if we will want more to know about osmotic pressure of a solution, it is equivalent to the pressure which must be exerted upon it to the prevent flow of solvent across the semi-permeable membrane. And therefore diffusion pressure deficit is equal to osmotic pressure minus Tp means the turgor pressure. Okay, and Tp is nothing but the wall pressure. Means after turgid of the cell, after turgidity of the cell, the pressure will form, that is known as turgor pressure, and it form onto the wall, onto the cell wall. So it is known as the wall pressure. Like, okay, now this diffusion pressure deficit is the osmotic pressure minus the wall pressure. So now see this osmotic pressure minus Tp is equal to Wp, means the turgor pressure is equal to wall pressure. So instead minus Tp, instead of Tp we can write here the Wp, that is the wall pressure. Now in flaccid cell means where cell will release the water outside the cell, the turgor pressure is 0. Obviously, when turgidity of the cell, turgor pressure is created onto the wall. But after shrinkage of cell, there is no turgor pressure. So, diffusing pressure deficit is equal to only osmotic pressure. Like this, here OP minus 0. So, dPd is equal to OP. Now, in turgid cell means when the cell will absorb the water and it form a pressure on the wall. So here the diffusing pressure deficit is 0. So turgor pressure is equal to the osmotic pressure. Now we will see about the facilitated diffusion. Now what is mean by facilitated diffusion? Facilitated, the meaning of facilitated means very easy. So it will make the protein carrier is present in this process, which will make the diffusion very easy. Now see why it is necessary. The passive absorption of solute when mediated by a carrier is called facilitated diffusion. Now see facilitated diffusion in a membrane showing ion channel and carrier protein. Now we will discuss about this diagram. Particles that are lipid soluble, this membrane is of lipid and protein. So lipid soluble cell membrane or lipoproteinaceous cell membrane, the diffusion of hydrophilic solute means the substances which are water loving solute has to be facilitated because their diffusion across the membrane is difficult. It is very difficult to pass the hydrophilic compounds into the cell. So membrane protein provide such sites for facilitated diffusion. Means to make the diffusion easy, the protein membrane is very important. So these protein are aquaporins, means these proteins are aquaporins, means it will pass the water molecule, and ion channels are here. See, this is protein channel or ion channel, means positive and negative charges will create here. This protein help move substances across the membrane. The substances which are present in extracellular space. So this protein will help to move these substances to pass these substances from this lipid proteinaceous membrane. And without expenditure of energy, means there is a no need or there is a no expenditure of the energy. Concentration gradient must be present for the molecule to be diffused through the facilitated diffusion. Now here the particles from extracellular space will enter into the proteinaceous carrier. This is the carrier, protein carrier. So it will take this, it will absorb this inside the protein carrier, and after enter this particle into this carrier, it will look like this, and then it will pass into the intracellular space of the cell in the cytoplasm. So this is our today's session. Now thank you very much. And again I am saying that please like, share and subscribe to our channel. Thank you.